Hello everyone. Today we're going to talk about viruses. Let's talk about the general characteristics of viruses. Viruses are obligatory intracellular parasites, meaning that viruses require a living host. They do not contain enzymes that would help them with their ATP production. They don't have the enzymes that will help them produce their own proteins, and so therefore they definitely need a host for them to survive and for them to replicate. Viruses either contain DNA or RNA, either single-stranded DNA or double-stranded DNA. Viruses, they contain a protein coat known as capsid. And so the capsid is protecting, or it's an outer covering that they have surrounding their DNA or RNA. Some viruses are enveloped viruses, so they actually have an envelope of protein on top of their protein coat. And some viruses, they don't have that extra layer, that extra envelope. Viruses require hosts because they are going to use the metabolic machinery and the enzymes of the host cell for them to be able to multiply and divide and basically survive. So the host range is definitely wide for viruses. Each virus, though, is really specific in terms of which host it's going to bind to, because viruses must actually attach to the surface of the host cell. They actually must chemically interact with the receptor, with receptor sites that are found on the surface of their host cells. And so, therefore, again, it's very specific because they need to have those specific attachment sites. Viruses that interact and attach themselves to bacteria, they're known as bacteriophages, or we usually say phages for short. So these phages, they're actually viruses that are able to stick to the surface of the cell wall of that bacteria, and then they are able to go ahead and infect the bacteria. And they would survive inside of that bacteria. And we're going to talk about the cycle of a virus living inside of a bacteria or living inside of an animal cell. We're going to talk about those cycles in just a few. Bacteriophages, they are actually pretty important when it comes to us humans, because we could use them to kill bacteria that are super resistant to a lot of the antibiotics that we have. A virion is a complete, fully developed infectious viral particle that's basically made out of nucleic acid, and the nucleic acid varies. It could either be DNA or RNA, and it could either be single or double-stranded, linear or circular. It's also going to contain a capsid, and the capsid is basically that protein coat, and it's made out of subunits called capsomeres. Some viruses have an envelope, and we call them enveloped viruses, and some do not. And that envelope is made out of lipids, proteins, and carbs. And the envelope, if the virus does have it, it's going to be surrounding the capsid. If a virus does have an envelope, usually also the envelope has spikes, which are like projections coming out from its outer surface. And each virus definitely has its own unique characteristics and shape. So right here, this is an image of a non-enveloped virus. So you can see the capsid right here, and you can see those subunits known as the capsomeres that are going to be surrounding the nucleic acid. So this is a non-enveloped virus right there. This is an image of an enveloped virus. If a virus has an envelope, then the envelope is going to surround the capsid of the virus, and it can have those spikes on its surface, which is basically a complex of proteins and carbohydrates. And those spikes are actually used to identify the virus. So each virus is going to have a unique shaped envelope and unique characteristics to it. When a virus infects a host, the host immune system is going to start producing antibodies against that virus. But some of these viruses can actually escape the antibodies that are produced by the host because viruses mutate very fast, and when they mutate, they actually change their surface proteins, and the virus would start looking different. And so the antibodies that were produced won't be effective against them because now the virus looks different, and so the antibodies are not going to be able to interact with them anymore. An example is the flu virus. So the flu virus is frequently undergoing changes and it's frequently mutating itself. So let's say that you've already produced antibodies against one flu virus and that virus mutates, it's going to look different and it can actually reinfect you. And that's why you get the flu shot every season, because every season the flu virus mutates and looks different, and so therefore, even though you've taken the flu shot last season, those antibodies that you've produced are not going to be effective against the newly mutated virus. This is an image of a bacteriophage, and we talked about bacteriophages. Those are viruses that infect bacteria. So you can see their capsid right there, and inside of it there is the nucleic acid. And usually bacteriophages, they have these tail fibers, and those tail fibers are what's going to attach to the surface of the bacteria. So here is the surface of the bacteria right here, and you can see that the tail fibers basically like stand on top of the bacteria. And again, this is due to the bacteriophages chemically interacting with these receptor sites that are found on the surface of the bacteria. So as we discussed previously, bacteriophages are viruses that can attack bacteria and kill bacteria. So if we have a plate right here, a nutrient agar plate with the bacteria, a lawn of bacteria growing on it, and bacteriophages were added to it, you will notice some clear plaques right here. And you can refer to them as just plaques or plaque forming units. Those clear regions right here are telling you that the bacteriophages were actually able to kill the bacteria living in this region. The region where you see no clear plaques would tell you that those bacteria were actually resistant, and the bacteriophage were not able to infect them or kill them. If we take a look at this graph right here, you see that during this period known as the eclipse period, the number of the virions are low. And that's because during this period, the virus is multiplying. It's making more of its DNA or RNA. It's multiplying its capsid. And so therefore the virus has not been assembled yet. You just have separate parts, the nucleic acid, the capsids, the proteins. And so therefore you can see right here when these parts start getting assembled, you can see that the virions, those complete infectious viral particles, are now being made, and all the parts are being assembled together. So you can see that as time passes, the virions are going to increase in number, and that's when you're going to have an acute infection because the number of virions inside of the host cell is really high. Now you might wonder, why is it decreasing? The thing is, as viruses increase inside of the host cell is going to die. We are going to talk about cycle known as the lytic cycle and how for these virions to actually get released from the host cell, they are going to burst out of the host cell. So therefore the host cell is going to die. And so if the host cell dies, then actually the virions are also going to start decreasing in number because, as we said, viruses require a host for them to survive. Right here, we're going to talk about the lytic cycle of a bacteriophage. Bacteriophages are the viruses that infect and attack bacteria. Because animal viruses, the cycle is different. So this lytic cycle right here has 5 main steps. First is attachment, the bacteriophage attaching to the cell wall of the bacteria. Second is penetration. The bacteriophage injects its own nucleic acid inside of the bacteria host. Third is biosynthesis, and that's when the virus is going to start using the host's enzymes and ribosomes to make its own viral components and to multiply inside of the host. 4 is maturation, and that's when all the viral components are going to be assembled together into virions. And then the last step is the bacteriophages that have been assembled inside of the host. They're going to break open the bacteria, and they are going to be released, and of course that's going to cause the bacteria to die. So let's go ahead and talk about it in detail. So here is the bacteriophage with its capsid. Inside you would find the nucleic acid of the bacteriophage. This right here is known as the tail sheath. And there is a little piece right there known as the tail core. You can see it better right there. There are also the tail fibers right here. So when the bacteriophage goes ahead and attaches, the very first step, to its host, what happens is the tail fibers go ahead and they interact and they bind to specific attachment sites on the cell wall of the bacteria. Once they go ahead and do that, the tail sheath contracts, kind of like a spring, and you can see it actually contracting right here. And then after it contracts, the tail core actually bores itself into the cell wall of the bacteria, and the nucleic acid gets injected into the bacteria. So kind of like a syringe, it goes ahead and injects the nucleic acid inside, and you can see right here, it's getting injected inside of the bacteria. How does it even penetrate? You might ask, that thick cell wall of the bacteria. Bacteriophages actually release through their tail fibers, they actually release enzymes known as lysozymes, and those lysozymes break down a portion of the bacteria cell wall. to make it easier for that tail core to basically drive through the cell wall and release the nucleic acid inside of the bacteria cell. Only the nucleic acid enters inside of the bacteria cell. So the capsid, the tail sheath, the tail fibers, all of these parts will remain outside the bacteria cell. They don't enter into the bacteria cell. Once the bacteriophage DNA is inside of the host's cytoplasm, the synthesis of the viral nucleic acid and the proteins, it's going to go ahead and begin. But what the virus does is it goes ahead and degrades, it breaks down the host's DNA. So if the host's DNA get broken down, basically the host, in this case the bacteria, cannot synthesize its own proteins anymore because its DNA has been damaged. Once the DNA of the host has been damaged, then the bacteriophage starts using the host's enzymes and to make copies of its own DNA, and it uses the host's ribosomes and amino acids actually to start going through the translation process, which is translating its own messenger RNA into proteins. So you can see that the bacteriophage is actually going to go through transcription and translation inside of the host, and it's basically using all of the host's resources from ribosomes, enzymes, amino acids. It uses it all so that it can make more of itself, make more of its own proteins, and make more of its own capsid, etc. And you can see right here, it's making more of everything, the sheath core, the capsid, the nucleic acid, the tail fibers. But notice how everything is not assembled yet. So right here, during the maturation process, this is when all of these components get together and they get assembled together. You can kind of see it right here in the bigger view, how all the parts get basically together and assembled. And once that happens, you get a complete virion. Then the plasma membrane of the bacteria cell is going to break open, again by viruses, by enzymes from the bacteriophage virions, and it's going to break open, and the bacteriophages, they are going to get released. Once these bacteriophages are released, they are actually able to go and infect other cells that are around, and this bacteria cell right here dies. So this is known as the lytic cycle, and again, lytic meaning bursting open. So you can see that the bacteria cell eventually it's going to burst open, and all of these bacteriophages are going to be released, and they can go infect other cells. Let's talk about the lysogenic cycles of bacteriophages. This picture right here is showing you a bacteriophage infecting an E. coli. And the bacteriophage that infects E. coli, we refer to it as the lambda phage. So the very first step is that bacteriophage needs to attach itself to the host. Once it does attach itself to the host, it starts to inject its own DNA inside of the bacteria host cytoplasm. You can see that the bacteria already has its own DNA right here. And now the phage DNA of the virus is now injected, and it can circulate inside of the cytoplasm of the bacteria. During the lysogenic cycle, a very interesting thing happens, and that is the phage DNA integrates itself within the bacterial DNA. So the phage viral DNA actually integrates and becomes attached to the bacterial DNA. And we start referring to it as the prophage. This prophage right here contains genetic information, and those genes could encode for several things. The virus might contain genes that encode for certain enzymes. The viral DNA could encode for some toxins. The DNA of the virus could contain, of course, genetic information, genes that encode for making new capsids, for making the tail fibers. Basically, they must contain genetic information to make all the parts of the virus. But do you see any new virions being assembled during the lysogenic cycle? No, you don't. And the reason why you don't see any new virions being assembled is because the genes that encode and they are responsible for synthesizing the capsid and the nucleic acid, etc., the tail fibers of the virus, those genes get turned off. And so therefore, no new virions are able to get assembled. The bacteria is going to go through its normal reproduction cycle. So it's going to be dividing and replicating itself. And as it does that, the bacteria needs to replicate its own DNA. But as it's replicating its own DNA, it's actually going to be replicating the prophage as well, because the prophage is now part of the bacterial DNA. And this can happen for several divisions. So as the bacteria is dividing, again, it's making copies of the phage DNA. Until that prophage, which is, again, the phage DNA integrated with the bacterial DNA, pops out. So basically, like, it cuts itself, it gets it removes itself from the bacterial DNA. What triggers that? Things like exposure to UV rays and exposure to certain chemicals. These events can lead to the excision or the popping out of the phage DNA. Once the phage DNA is it's now not integrated anymore with the bacterial DNA, it can actually enter into the lytic cycle. So when it enters into the lytic cycle, the genes that are responsible for synthesizing the capsid and the tail fibers, etc., those are not turned off anymore, and therefore you see new virions being assembled, and the parts of the virus is actually being made and assembled and everything, and the bacteria is eventually going to lyse, it's going to burst open, and the bacteriophages are going to be released. Once they are released, they are going to go ahead and infect other cells that are around. Now, the lysogenic cycle results in two important things. One is known as phage conversion. What is phage conversion? We said that during the lysogenic cycle, an interesting event takes place, and that is the viral DNA, the phage DNA, integrates itself with the bacterial DNA, resulting in that prophage. Now, as I said, the viral DNA contains many genes, and some of these genes actually code for toxins. So some bacteria are now going to have new characteristics. Why? Because let's say this bacteria did not have the gene that encodes for releasing the toxin, a specific toxin, but now because it has this prophage carrying the gene that releases this specific toxin, now the whole entire bacteria can actually release that toxin. An example of certain bacteria that would take advantage of these prophages is Clostridium botulinum, which causes botulism. These bacteria actually are able to cause botulism because of the prophage gene, because the prophage gene is actually the one carrying the gene that will release that specific toxin. And so there are so many other bacteria that again are only going to be pathogenic really, and they're only going to cause diseases when they have a prophage within them, because the prophage is the one responsible for synthesizing those toxins. So without this prophage gene, those bacteria would not be considered pathogenic. And so that's an important thing known as phage conversion that takes place during the lysogenic cycle. The second important result of the lysogenic cycle is something known as specialized transduction, which we are going to talk about right now. We said that the lysogenic cycle results in two important things. One is prophage conversion and the other one is specialized transduction. So let's talk about specialized transduction. Let's say that here is a bacteria and the bacteria containing its own DNA right there. Let's say that this bacteria contains a gene known as the GAL gene. GAL is short for galactose. So this bacteria has the ability to break down and metabolize galactose. Not all bacteria are able to metabolize galactose, by the way. Let's say that this bacteria gets infected by a virus and the virus went ahead and started the lysogenic cycle and it integrated its own phage DNA with the bacterial DNA, and here now you have the prophage. If this bacteria gets exposed to certain chemicals or to UV light, the phage genome, right here, this pink one, this phage genome gets cut off from the bacterial DNA. But what happens is part of the bacterial DNA could actually stay behind attached to the phage DNA. So now you have the phage DNA with a piece of genetic information from the bacteria host. So let's say that the part that was removed from the bacteria host DNA was the GAL gene. So now this phage DNA is carrying the genetic information needed to break down galactose. After this happens, after the excision of the phage DNA takes place, the virus is going to start using the host's ribosomes and enzymes, etc., to start assembling its parts, its capsid, its tail fiber, etc. And as you can see, new virions are going to be assembled. And you can see that this bacteria host right here, well, it's going to die because it lyses open and the virions are released. Once the virions are released, they can go ahead and infect other bacteria hosts. So let's say here is a new bacteria host, but what's interesting about this bacteria is its DNA doesn't contain the genetic information needed to break down galactose. So that's why we are saying that this bacteria right here is galactose negative, because it doesn't have the genes needed to encode for the proteins, etc., that would help this bacteria break down galactose. But let's say that this bacteria right here that is galactose negative gets infected by a bacteriophage. And the bacteriophage, as you can see, attaches itself and starts releasing its phage DNA. But now this phage DNA is not only viral DNA, it's viral DNA and a piece of the old host's DNA, which is the gene that's going to help metabolize galactose. Now, if this cell enters into the lysogenic cycle, what happens is the phage DNA integrates itself with the host's DNA. And now you have a prophage. So now this bacteria host is now going to be able to metabolize and break down galactose. Why is it going to have that new ability? Because it now has this information integrated with its own DNA, and so therefore it is going to now have the ability to break down galactose. So specialized transduction, if we want to summarize really what's it all about, specialized transduction is the ability for the virus to actually carry a piece of the old host's DNA and integrates it into new hosts. That's what special transduction is all about. Prophage conversion is the idea that the host is now going to carry new genetic information that it didn't carry before. And in this case, the new genetic information that this bacteria has gained is the ability to break down galactose. Now, animal viruses are different. Animal viruses, when they attach themselves to a host cell, again, they do have to bind to specific attachment sites, but animal viruses usually don't have those tail fibers that bacteriophages have, so they look different compared to bacteriophages. And then how they enter the host cell is also different. They could either enter by a process known as receptor-mediated endocytosis or fusion, and we'll talk about the two. But when they enter into the host cell, the whole virus enters, meaning that the capsid is also going to be entering into the host cell, versus with bacteriophages, as we remember, the bacteriophage just sits on the surface of the host cell and injects just the DNA, and then everything else stays outside of the host, the capsid, the tail fibers, etc. Animal viruses, they're different. The whole thing enters inside of the host. And so therefore, because the capsid enters into the host cell, it has to go through a process known as the uncoating process, because the capsid must break open so that the viral DNA can get released inside of the host. And this uncoating process, the virus could either use its own viral enzymes or basically the host enzymes to help with that process. Then the virus is going to start producing and making its own nucleic acids and its own proteins, of course, with the help of using the host's enzymes and ribosomes, etc. And it's going to start to assemble itself. Now, the way it gets released out of the host is also a bit different. It could go through something known as budding, and that's specifically for viruses that have an envelope. The viruses that have an envelope, they will actually bud out of the host, and they actually take a piece of the host, a part of the plasma membrane of the host, and that's going to become the envelope basically for the virus. Or it could rupture out. So we will take a look at all of these steps. Animal viruses enter into their host cells in a different way compared to bacteriophages. So as we said, bacteriophages, they use their tail fibers and they stand on top of the host cell and then they inject their DNA. Animal viruses enter the host cell in completely different fashion. There are two ways they enter the host cell. One is known as endocytosis and one is known as fusion. During the endocytosis process, the plasma membrane of the host, so they're highlighting it to you right here by that pink color, the host plasma membrane starts to dip inwards right here. You can see that it's starting to invaginate basically, and it will start basically enfolding the whole entire virus. So it enfolds the virus and the virus basically gets sucked into the host cell. The whole thing, so the capsid of the virus and its nucleic acid gets pulled into the host. A different way of entering into the host cell here is the plasma membrane, one more time, of the host cell. The envelope of the virus could actually integrate itself with the plasma membrane of the host, and the capsid and the nucleic acid of the virus gets sucked into the host. So this process is known as endocytosis or receptor-mediated endocytosis, and then this one right here is known as the fusion process. And in both cases, the virus capsid is going to enter into the host, and so therefore animal viruses, after they get sucked into the host cell, they have to go through a process known as uncoating, and that is the capsid needs to break open so that the viral nucleic acids could actually be released into the host. Now, when viruses are leaving the host cell, they could leave in two different fashions. One is the host cell could rupture open, kind of like what happens during the lytic cycle. So the host cell is going to rupture open and the virus is going to be released. And that's definitely going to cause the host cell to die because it's rupturing open. A different way for the virus to actually leave the host cell without killing it. So it leaves it, but it doesn't kill the host cell. That process is known as budding. So here is the host cell, and here is the viral capsule with its nucleic acid inside of it. What happens is the host plasma membrane starts to dip, as you can see, and it gets larger and larger, and the virus enters into that enfolding that's happening right there, and it completely detaches. And actually, this piece now of the host plasma membrane is actually going to become part of the envelope of the virus. So part of the envelope of the virus is actually going to be part of the host's plasma membrane. So you can see right here the plasma membrane starts to dip and the virus enters into it, and then it completely gets detached off. That's usually how enveloped viruses leave the host cell. So enveloped viruses, they leave via that process known as the budding process. So if we do a quick comparison between bacteriophages and animal viruses, bacteriophages, we know that when it comes to the attachment process, they use their tail fibers to stand on the host, and then they inject their DNA. Animal viruses, they don't have tail fibers, so they are still going to attach to specific sites on the plasma membrane of their host, but again, they don't have these tail fibers. Bacteriophages, they're going to inject only their DNA into the host cell. So the capsid and the tail fibers, all of these will remain outside of the host. Versus with animal viruses, the capsid with the nucleic acid enters into the host cell, as we said, either via the process known as the receptor-mediated endocytosis or via the fusion process. Because the bacteriophages only inject the DNA inside of the host, the process known as uncoding is really not necessary. It's not required at all. But because animal viruses, the whole virus, including its capsid, enters into the host, that capsid has to come off. So it has to go through the uncoding process. Once the DNA of the bacteriophage is inside, it's going to stay within the cytoplasm of the host cell, and from there it's going to start either entering into the lytic cycle or the lysogenic cycle. With animal viruses, if the animal virus contains DNA, then the DNA usually enters into the nucleus of the host. But if it's an RNA virus, then it's going to stay within, or it's going to enter first into the cytoplasm of the host. During the lysogeny cycle of the bacteriophage, we talked about how the bacteriophage or the phage DNA integrates itself within the bacterial DNA, and then once it gets excised off of the bacterial DNA, it can enter into the lytic cycle, and the host cell usually dies because it ruptures, opens, and the bacteriophages are released. With animal viruses can become latent, so they are not going to be expressed, and they kind of stay dormant inside of the host cell, and they can cause like very slow viral infections because they are pretty slow in terms of their replication process. Some viruses can actually lead to cancer. So they can actually cause the host cell to replicate rapidly, and they can actually turn it into a cancerous cell. Enveloped viruses, for them to be released, they need to bud out. Non-enveloped viruses, they usually just cause the host cell to die because the host cell is going to rupture to release those non-enveloped viruses. The budding process usually doesn't kill the host cell. So let's say here is a virus, here is the capsid of the virus, and it contains nucleic acid, and let's say that this nucleic acid is DNA. This is an animal virus, so it's not going to have the tail fibers like bacteriophages. It's still going to have to attach to specific attachment sites on the plasma membrane of the host. As I said, when it enters, it could either enter via the process known as the receptor-mediated endocytosis or fusion. Depending on whether it has an envelope or not. Usually viruses that have envelopes, they will enter via the fusion process because their envelope is going to integrate itself with the plasma membrane. But either way, whether it's the fusion process or whether it's the endocytosis process, the capsid is also going to enter into the host. Then comes the uncoding process. So the capsid must break down and the viral DNA is going to be released. Once the viral DNA is released, it's actually going to enter into the nucleus of the cell. So here is the nucleus of the host, and the viral DNA enters into the nucleus of the cell, and it's going to start using the enzymes of the host, and the DNA is going to be transcribed into messenger RNA. And again, it's going to continue using the host's enzymes and basically resources to go through translation, and it's going to start making its proteins, making its capsids, etc. So you can see that the virus is going to start making its own proteins and capsids, and you can see right here the capsid proteins, they are being made. The the viral proteins are also being made, and then the maturation process is going to take place. So the viral components right here, they're going to start getting assembled, and new virions are going to start maturing, and the virions are going to be released. Now, they're going to be released via two ways. Either the host is going to rupture open, and that's going to usually cause the host cell definitely to die because it's rupturing open, and the virions are going to be released, or as you can see in this case, it's actually budding, so the plasma membrane enfolds itself around the virus, and part of the host's plasma membrane becomes part of the envelope of the virus. And again, usually viruses that have envelopes, that's how they get released. But if it's a virus that doesn't have an envelope, then what usually happens is the host cell ruptures open and the virus gets released. And then, of course, once the virus is released, it can go ahead and infect another cell. Some viruses are known as retroviruses, and that's because those viruses contain RNA for their nucleic acids, and they contain an enzyme known as reverse transcriptase. Reverse transcriptase is an enzyme that will convert the nucleic acid, the RNA nucleic acid of a virus, into DNA, into viral DNA. And that's why we call it reverse transcriptase, basically meaning like reverse transcription. Transcription is when the DNA is transcribed into RNA, and in this case, the virus is using certain enzymes known as reverse transcriptase to basically reverse its RNA and make DNA out of it. So you can see right here, here is a retroviruses getting sucked into the host cell. So first it has to attach to the host cell plasma membrane, then it gets pushed inwards. And again, we talked about how it gets into the host cell, either by the fusion or by the receptor-mediated endocytosis. Once it enters into the host cell, the capsid will have to be uncoated, so using some enzymes from the host, or again, enzymes carried by the virus, the capsid is going to break open, and as we said in this case, the RNA inside of that virus is going to be turned into viral DNA by using the reverse transcriptase enzymes. The viral DNA is going to enter into the host's nucleus, and it's actually going to integrate itself with the host's DNA. And once it integrates itself with the host's DNA, we're going to call it the provirus, kind of like the prophage when we were talking about bacteriophages, integrating their DNA with the bacterial DNA, and we ended up calling it a prophage. A provirus is really the same thing. The viral DNA is now a part of the host's DNA. But the biggest difference between a provirus and a prophage, we talked about how prophages can actually be excised off the bacterial DNA when the bacteria is exposed to UV light or certain chemicals. A provirus doesn't ever gets excised off. So provirus is always going to be part of the host's DNA, and it's never going to be cut off from the host's DNA. Viruses that actually integrate their DNA with the host's DNA can lead to cancer, because when a virus integrates its own DNA, that's going to alter the host DNA, and that can lead to cancer. Actually, 10% of cancers out there are actually due to a viral infection. So once the provirus right here is now part of the host's DNA, it can use the host enzymes and machinery to basically start going through its own transcription and translation and making its own proteins and making its capsid, etc. And the viral proteins and capsids, they're going to be assembled together, and you can see new retroviruses being assembled, and they will bud out of the host cell, and you can see that the plasma membrane of the host cell buds out, and part of it is going to become part of the envelope of the virus. And once the virus is released, if it's released via the budding process, that's usually not going to kill the host cell. And once it's released, it can go ahead and infect a new cell. So retroviruses are known, some of them are known to actually cause cancer. So that's because, again, they integrate their DNA into the host's DNA, causing changes to the host's DNA, and again, that can lead to cancer. So this is an example of how animal viruses, specifically retroviruses, go ahead attached to their host cells and how they integrate. First, they have to reverse their RNA into DNA, and then the DNA integrates itself with the host's DNA, and it becomes part of it, and the cycle continues.